Hello and welcome everybody to my channel Omni Bioentrance, and today we will be seeing West Bengal State Eligibility Test for the subject Life Science, paper 2, conducted in the year 2020. So coming to the first question, identify the incorrect statement regarding lipid rafts. So lipid rafts, they are enriched in class of integral membrane protein, and these are anchored to the membrane by two covalently attached long chain saturated fatty acids and glycosyl phosphatidyl inositol. So the incorrect statement here is membrane proteins are totally absent in the lipid raft region. That is incorrect. Then lipid rafts are involved in important cellular processes including endocytosis, signal transduction and chlorophyll tracking. That is correct. Lipid rafts are primarily composed of cholesterol and sphingolipids. That is also correct. Lipid rafts are organized membrane microdomains. That is also correct. The incorrect answer is option D. Then next one. Why are the haploid plants induced through pollen culture technique suited for mutation research? So pollen culture, it is a technique of production of haploid plants by the culture of pollen grains obtained from the anther of the plant, and it is done under aseptic conditions and inoculated into an artificial media of known composition. And why are the haploid plants induced through pollen culture technique suited for mutation studies? Because pollen itself is haploid and it is a single cell system. So the answer here is they allow immediate expression of recessive mutation. Since it is haploid, you can easily study the various recessive mutations. So answer is option C. Then tetrasolium test in pollen viability is based on the activity of which of the following enzyme? The most commonly used tetrasolium salt in pollen viability test is 2,3,5-triphenyl tetrazolium chloride. And tetrasolium test, it is based on the reduction of soluble colorless tetrasolium salt to reddish insoluble formazan in the presence of dehydrogenase. So if the pollen grains are incubated in tetrasolium solution, viable pollen grains take a reddish color due to the formation of formazan by dehydrogenase action. So the enzyme used here is dehydrogenase. Answer is option B. Then next one. Red rust of tea is a common disease is caused by which of the following? So red rust of tea is caused by Cephalourus virescens. Answer is option A. Then next one. Succinate dehydrogenase converts succinate to fumarate. Which one of the following is true when the competitive inhibitor malonate is added to the reaction? So competitive inhibitor, they increase the Km value and decrease, and the Vm remains unchanged. So the answer here is option C. Km increases, Vmax remains unchanged. Then in an enzyme catalyzed reaction, the competitive inhibition by an inhibitor is mediated when it binds at several sites on an enzyme, binds reversibly at the active site of an enzyme. Yes, that is correct. It binds reversibly at the active site of an enzyme. Then next one. What are amino acids catabolism? Where does amino acid catabolism predominantly occur? So it occurs in the liver. Answer is option C. Then the full form of MALT is, it is mucosa associated lymphoid tissue. So answer is option B. Then next one. The large granular bone marrow derived non-phagocytic lymphocytes capable of recognizing and killing virally infected and neoplastic cells they are called as NK cells or natural killer cells. Answer is option B. Then next one. Each individual antigenic determinant of the variable region of an antibody is referred to as. So the first option is paratope. So paratope is a part of an antibody which recognizes and binds to the antigen. So this is the paratope. Then epitope is also called antigenic determinant, is a portion of the antigen that is capable of stimulating an immune response. This is the epitope of an antigen. Then agritope is the region of the peptide that interacts with the MHC molecule. And idiotypes, these are markers. See, these are the markers. These are idiotypes. These are markers on the variable domain. This is the variable domain of antigen-specific immunological receptors recognized by anti-idiotypic antibodies or T cells. So these are the idiotypes. So what they have asked, each individual antigenic determinant of the variable region of an antibody is referred to as, they are referred to as idiotopes. So answer is option D. Then next one, numerical taxonomy, which involves the numerical evaluation of the similarity between taxonomic units and ordering of these units into taxa on the basis of their similarity, was coined by. So it was coined by Sokal and Smith. Answer is option B. The genome of rotavirus consists of, it consists of a double-stranded RNA. Answer is option D. The next one, the symbiotic association of nitrogen-fixing prokaryotes with higher plants often result in nodule formation. Which of the following association represents an exception? So the answer here is Anabena and Azolla. It is a sample of Azolla will form filaments of, a sample of Azolla will have filaments of Anabena living within ovoid cavities inside the leaves. All the other forms, they form nodules except Anabena Azolla symbiotic association. Then next one, when growing on agar, most members of the group of bacteria form colonies with fried egg appearance. So it is, answer is option D, that is molecules. They do not have cell wall and form fried egg appearance. Answer is option D. Then next one, highly repetitive DNA sequences are normally concentrated in eukaryotic chromosome near its centromeric region. That is the region where highly repetitive DNA sequences are normally found on a eukaryotic chromosome. Then next one, which of the following pair of protein regulate production of anterior structures in Drosophila embryos? So it is bicoid and hunchback. Answer is option B. Bicoid, it regulates expression of gene responsible for anterior structures as well as it stimulates hunchback. And hunchback, it regulates transcription of genes responsible for anterior structures. So the answer here is bicoid and hunchback. Then next one, which one of the following components is not engaged in association of actin filaments with Z disc during muscle contraction? So it is vinblastine. Vinblastine is vinca alkaloid used in the treatment of cancer. So that is not involved. So answer is option D. Then next one, during synchronous contraction of cardiac muscle cells, electrical impulses is transmitted through. So electrical impulses are transmitted through gap junctions. Because gap junctions, they are responsible for electrical coupling and transfer of so small molecules between cells, whereas desmosome-like junction provides strong intercellular adhesion. Tight junctions don't allow transport. The next one, vinblastine is an important alkaloid, is extracted from which of the following plants? Vinca rosea or Catranthus roseus. Answer is option D. Then evolutionary changes in one species prompt corresponding changes in other species with which of the following with which the former interacts ecologically. The process is known as. So evolutionary change in one species prompts corresponding change in other species with which the former interacts ecologically. This process is known as. So first one is we will see microevolution. Microevolution, it is defined as changes in the frequency of a gene in a population. What is parallel evolution? It occurs when independent species acquire similar characteristics while evolving together at the same time in the same ecospace. And what is genetic drift? It is change in the frequency of an existing gene variant in a population due to random chance. So what is left out? Coevolution. So coevolution is the answer. It is evolutionary change in one species prompts corresponding change in the other species with which it is interacting ecologically, and this process is known as coevolution. Then next one, porogamy refer to as. So it is fertilization in which pollen tube enters the ovule through the micropyle. Answer is option D. Fertilization in which pollen tube enter the ovule through the micropyle. Then next one. Following are four considerable modes of inheritance for the given pedigree: X-linked recessive, X-linked dominant, autosomal recessive. Which of the above modes of inheritance can explain the pedigree shown below? So they have given the pedigree analysis over here. So if you have capital A small a, that is heterozygous condition, then you will get these following gametes. So following are four considerable modes of inheritance for a given pedigree. Which of the above mode of inheritance can explain the pedigree shown below? X-linked recessive means the males should have got the disease. So autosomal dominant means here at least 3 will have the particular trait and 1 will not have the dominant trait. So it is autosomal dominant. Answer is option C. Then next one, deamination of guanine produce. So it produces xanthine. Answer is option B. Then which of the following enzyme dissociates HCO3 minus ions into carbon dioxide and OH minus, that is hydroxyl ions, in pulmonary capillaries, that is carbonic anhydrase? Answer is option D. Then consider a bacterial cell performs anaerobic respiration. If the bacterial cell had access to 20 molecules of glucose, how many molecules of ATP will be produced? So it is anaerobic respiration and it has access to 20 molecules of glucose. So in anaerobic respiration, 1 molecule of glucose produces 2 ATP molecules. So 20 molecule produce 20 into 2, that is 40 molecules. Answer is option D. Then phosphatidylserine, an important component of the biological membrane, is located in. So it is located in the inner leaflet but flip-flop to outer leaves at specific condition. Answer is option D. Then configuration of a protein can only be altered by breaking disulfide bonds. Answer is option B. Pollination of flower by bat is termed as. It is termed as chiropterophily. Answer is option A. Melitophily is pollination carried out by bees. Myophily is by flies and anemophily is by wind. The next one, which of the following hormones initiates biological action by crossing the plasma membrane and then binding to a receptor? So steroid hormones, they are made of lipids, that is phospholipids and cholesterol, and they are fat soluble, and so they can easily cross the plasma membrane of the target cell and bind to receptors in the cytoplasm of the cell. So here the answer is estradiol because it is a steroid hormone. Answer is option B. Then next one, the process by which cells secrete macromolecules by fusing vacuole membrane to the plasma membrane is called. So it is called endocytosis. Endocytosis is a cellular process in which substances are brought into the cell. The material to be internalized is surrounded by an area of cell membrane which then buds off inside the cell and form vesicle containing the ingested material. And endocytosis, it includes pinocytosis, that is cell drinking, and phagocytosis, that is cell eating. So what they have asked here is the process by which a cell secrete macromolecules. So it is secreting macromolecules by fusing vacuole membrane to the plasma membrane. It is called as exocytosis. Answer is option B. Then colchicine is an inhibitory chemical which prevents. So colchicine, it prevents microtubule polymerization. Answer is option A. Then which of the following plants are snail pollinated? So they are aroids. Answer is option A. Then nori, a highly priced algal food, is obtained from. It is obtained from red alga porphyra. Answer is option A. Then woods of cycads are mostly belong to which of the following types? So manoxylic wood, it is a gymnosperm wood and it has low number of xylem cells in them, and it has non-compacted wood with large pith cell. Then the stem of cycas, it is monoxylic when young, that is it has only a single ring of vascular bundles, but after secondary growth it becomes polycylic, that is it has many rings of secondary vascular tissue. So the wood of cycads is monoxylic as well as polycylic. So answer is option C. Then in which of the following years APG4 has been published? So it is angiosperm phylogeny group. Answer is option B, that is 2016. Then alternative name of albuminous cell is. So it is also called Strasburger cells of albuminous. It is also called Strasburger cell. Answer is option C. Then which of the following is not part of the endocrine system? So the pancreatic acinar cells, that is the acini of the pancreas, they are not part of the endocrine system. It is a functional unit of the exocrine pancreas and it synthesizes, stores and secretes digestive enzymes. And the primary function of this acinini is to synthesize and secrete hydrolytic enzymes into the duodenum for digestion of food. Then next one. Collagen instability and connective tissue abnormalities are caused due to the absence of. So they are caused due to the absence of vitamin C. Answer is option B. Then next one. The protein products encoded by two homologous genes that are found in different species are called. So orthologs. Orthologs are genes in different species that evolved from a common ancestral gene by speciation, and orthologs, they retain the same function during the course of evolution. That is, they encode proteins with the same function in different species. Then what are paralogs? They are homologous genes that have evolved by duplication and code for protein with similar but not identical functions. Then what are homologs? It is a type of gene that is inherited by two different species that evolved from the same ancestor. Such as if you see in the case of house cat and leopard, they share a common ancestor. What are analogs? Analogous genes are genes that have identical or similar functions but don't share common ancestor and are therefore unrelated. So the question was, the protein products encoded by two homologous genes that are found in different species are called. They are called orthologs because orthologs are genes in different species that have evolved from a common ancestral gene. So the answer here is option A, that is orthologs. Then frame shift mutation is caused by the agent. So they are caused by intercalating agent. So here example is intercalating agent is proflavin. Answer is option C. An intercalating agent, they are of the same size as a nucleotide and they produce mutation by intercalating between adjacent bases in DNA and thereby distorting the three-dimensional structure of the DNA and causing single nucleotide insertions or deletions during replication. And these insertion and deletion, they can lead to frameshift mutations. That is the effect of intercalating agents. Then EMS is alkylating agent, nitrous acid is deamination of cytosine to uracil, and 5-bromouracil is a base analog. So the answer here is proflavin. Then next one. A chromosome locus is represented by two alleles, capital A and small a. The frequency of capital A in the population is 200 individuals is 0.3. The expected number of heterozygotes in the population will be, so P is equal to 0.3, P plus Q is equal to 1, Q is equal to 0.7. Frequency of heterozygotes will be 0.42. That is 42 in 100 individuals. So in 200 it will be 84. So the answer is option C. The next one. If the husband carries an X-linked deadly disease and wife is homozygous normal, select the correct option from the following. So they will have a 50% chance of having the affected male child. The husband is having the X-linked disease, whereas the wife is normal. So chances of male having the disease is very less. It will not, there will not be any 50% chance of having the disease. Then they can safely go for only a female child. They can safely go for only a male child. They should not plan to have a child. So here the husband is only the carrier of the disease, is having the disease, whereas the female is normal. So the female child which are born to these people, they will be carriers of the disease. Okay, because it is X-linked disease, and we cannot say that they can go happily for a female child, because female child will have a tendency to spread the disease to the next generation. Then they can safely go for only male child. That is correct, because the male child will not be affected by the disease. They should not plan to have a child, that is also wrong. And they will have 50% chance of having affected male child. Male child getting affected is not possible with this kind of cross. So the answer is they can safely go for only male child. Then which one of the four of the cranial nerves have both sensory and motor function? So that is facial. Answer is option A. Then in testis and sperm cells, fructose is transported through the transporter. So GLUT2, it is a facultative glucose transporter located in the plasma membrane of liver, pancreas, intestine, kidney cells, and as well as in hypothalamus area. Then GLUT3, it is a major neuronal glucose transporter present in both dendrites and axon. Then GLUT4, it is insulin regulated glucose transporter found primarily in adipose tissue and skeletal muscles, as well as in cardiac muscles also. Then GLUT5, it is a high affinity fructose transporter primarily expressed at the apical surface of intestinal epithelial cells, but also at lower levels in testis, kidney, brain, skeletal muscle and white adipose tissue. So they have asked in testis, sperm cells, fructose is transported through the transporter. So it is GLUT5. Answer is GLUT5 is the fructose transporter. All the others are glucose transporters. So answer is option D. Then which fish species does perform catadromous migration? So catadromous fish spend most of their lives in fresh water, then migrate to the sea to breed. So eels of the genus Anguilla is an example of catadromous fish. So it is answer is option C, Anguilla. It is called European eel. Then next one. Chloroquine is used as an anti-malarial drug against the malaria caused by protist Plasmodium falciparum. Resistance against chloroquine occurs in the protist due to amplification of the gene encoding ABC transporter that pumps out the drug from the cytoplasm. That is the answer. Answer is option A. Then next one. A tautomeric shift causing the substitution of one purine for a pyrimidine is called. So the version has changed. That is, purine has become pyrimidine. So it is transversion. Answer is option A. If it is transition, then purine will change to another purine, or pyrimidine will change to another pyrimidine. That is transition. Here version itself has changed. That is, purine has changed to pyrimidine. So you can remember it like that. Version change, transversion. Then next one. Which of the following agents stimulate the heart and inhibits the ability of the heart directly to relax? So it is sympatholytic. It is not sympatholytic, it is sympathomimetic. Okay. Answer is option B. Sympathomimetic. Sympathomimetic drugs are stimulant compounds which mimic the effects of endogenous agonist of the sympathetic nervous system. It increases heart rate, force cardiac contraction. So which of the following agents stimulate the heart and inhibit the ability of the heart directly to relax? So they are sympathomimetic drugs. The next one. Which of the following protein links the actin to intermediate filaments in a cell? So it is plectin and spectrin. So both B and D are correct. Answer is option B as well as option C is also correct. Not C, option D is correct. Spectrin and plectin. Then next one. The smallest three-dimensional volume of soil which gives full representation of horizontal variability is called. It is called as speedon. Answer is option B. Soil horizon is a layer parallel to the soil surface whose physical, chemical and biological characteristic differs from the layers above and beneath. Then what is topography? It is the study of forms and features of land surfaces. So the answer here is pedon. Then which of the following statement about Giardia lambelia is not correct? So Giardia lambelia, it has both trophozoite and cyst stages in its life cycle. That is correct. Then it is transmitted by fecal oral route. Yes. Then it causes hemolytic anemia. That is not correct. It is the common cause of diarrhea in humans and other mammals throughout the world. So it can be diagnosed by string test. That is also correct. So it causes hemolytic anemia is not correct. Answer is option is not correct. Okay, they have asked for not correct. So it causes hemolytic anemia. That is not correct. Then next one. Antibacterial antibiotic that binds to plasma membrane and disrupts its structure and permeability properties is that binds to plasma membrane and disrupts its permeability properties is. So here tetracycline, it is an inhibitor of protein synthesis, and vancomycin, it acts by inhibiting proper cell wall synthesis in gram-positive bacteria. And it targets the D-alanine terminus of the peptidoglycan. Then bacitracin, it is a complex polypeptide produced by Bacillus subtilis, and it inhibits peptidoglycan synthesis during the second step of bacterial cell wall synthesis by interfering with the activity of phosphorylase, and it is bactericidal. Then you have polymyxin. So polymyxin B is an antibiotic that inhibits bacterial membranes after binding to the lipopolysaccharides, interacting with the lipid A of the outer membrane and acting as a detergent by disrupting the membrane phospholipids, causing cellular permeability. So polymyxin antibiotic, it inhibits bacterial membranes after binding to lipopolysaccharides. Then it will interact with the lipid A on the outer membrane, and it also acts as a detergent by disrupting the membrane phospholipids, causing cellular permeability. So the answer here is polymyxin B. Then which of the following inhibitors of electron transport system is a secondary metabolite of Streptomyces species? So it is antimycin A. Answer is option C. Then the agitator or impeller in a stirred fermenter do not carry out the function of. So they do not carry out the function of mechanical form breaker. Answer is option D. Then next one. Which of the following antibiotics resemble penicillin in terms of their structure and mode of action? So it is cephalosporins. Answer is option C. Then which of the following pairs of immunoglobulin isotypes have characteristic J chain to form the polymeric structure? So it is IgM and IgA. They are having the characteristic J chain. Answer is option B. Then macrophages express MHC class 2 after getting activated by interferon gamma secreted from which one of the following? That is C cells, that is CD8 cytotoxic T cells. Answer is option A. Then a molecular cloning vector should have all of the following except. So it should have a multiple cloning site, yes. Autonomously replicating sequence, yes. Antibiotic resistance marker sequence, yes. Reverse transcriptase, that is not required. So answer is option D. Then aminopeptidase catalyzes the hydrolysis of amino acids from the N-terminal end of a protein, while carboxypeptidase catalyzes the hydrolysis of amino acids from the C-terminal end of a protein. Chymotrypsin catalyzes the hydrolysis of peptide bonds following aromatic amino acids, while trypsin catalyzes the hydrolysis of peptide bond following lysine and arginine. Select the correct order from the following. I don't know whether you can see the 1, 2, 3 marked over there. Here is 1, this line is 1, this line is 2, this line is, okay, this line is 1, this line represents 2, this line is 3, and this line is 4. So we have to see which one. So here they have told aminopeptidase catalyzes the hydrolysis of amino acid from the N-terminal end of a protein. So 1 will be naturally aminopeptidase, that is the N-terminal end of the protein. So answer is 1. 1 is aminopeptidase. Then they have told while carboxypeptidase catalyzes the hydrolysis of amino acid from the C-terminal end. So 4 is carboxypeptidase. This is the C-terminal end. So it will cut over here. So 4 will be carboxypeptidase. Then chymotrypsin catalyzes the hydrolysis of peptide bond following aromatic amino acid. So this is an aromatic amino acid. So the chymotrypsin, it will cleave over here. So 2 is chymotrypsin. And then trypsin catalyzes the hydrolysis of peptide bonds following lysine and arginine. So this is the lysine, I think, or lysine, I think. So it will cleave over here. C-terminal end of lysine residue will be cleaved over here. So 3 will be trypsin? Answer is option A, that is 1 aminopeptidase, 2 chymotrypsin, 3 trypsin and 4 carboxypeptidase. So answer is option A. Then in eukaryotic cell, the assemblage of ribosomal subunits take place in the nucleoli. Answer is option A. Then next one. The most suitable in vitro technique considered for production of genetically stable clones of plants is, so shoot meristem culture. That is correct. Okay, callus mediated organogenesis can give rise to soma-clonal variation, so that cannot be considered. Then callus mediated somatic embryogenesis, that also cannot be considered. There can be soma-clonal variations in that also. Then androgenesis, it is haploid plant production, so that is also not possible. So the most suitable in vitro technique considered for the production of genetically stable clones of plant, it will be shoot meristem culture. Then next one, heterokaryons generated through protoplast fusion can be quickly detected by. So they can be detected by fluorescein isothiocyanate. Answer is option C. And protoplast fusion, it permits the production of somatic hybrids. One cell can be induced to fuse with another cell to form the heterokaryon, which consists of cytoplasm containing chloroplast, mitochondria and nuclei from both the parent cell. That is heterokaryon. Okay. So it contains cytoplasm, chloroplast, mitochondria and nuclei from both the parent cells. That is a heterokaryon. So it can be quickly detected by fluorescein isothiocyanate. Then next one, in particle gun direct gene transfer technique, the size of the microprojectile gold particle ranges from 1 to 3 microns. Answer is option B. Then next one, what is the minimum loss of original habitat which is necessary to designate a region as biodiversity hotspot? That is 70%. Answer is option C. Then which one of the following is the limitation of liposome based drug delivery system? So liposomes, they are spherical vesicles consisting of one or more concentric phospholipid bilayer enclosing an aqueous core. So they are non-toxic and biodegradable, and they can be used as powerful delivery system for several drugs. So which of the following is a limitation of liposome based drug delivery system is they are biocompatible. Option A is correct. Then they can entrap both hydrophobic and hydrophilic drugs. That is also correct. They undergo structural changes over time during storage and lead to breakage of drug molecules. That is not correct. They don't lead to breakage of drug molecules. And they are tolerant to extreme pH conditions. That is also correct. So it is not is the limitation of yeah, that is possible because they can lead to breakage of drug molecule, and that is limitation of liposome based drug delivery system. So answer is option C. So C is the correct answer. I made a mistake over there. It is a limitation of liposome based drug delivery system. Answer is option C. Then next one. Which of the following statement is not true regarding the plant growth hormone brasinosteroids? So brasinosteroid promote cell elongation and cell division. Yes. They facilitate differentiation of xylem tissues. Yes. They inhibit leaf abscission. Yes. They are often induce dwarfism in plants. They often induce dwarfism in plants. No, they promote cell elongation, so dwarfism cannot be induced. So the answer here is option D. Then in analysis of variance one-way model, it is observed that the value of the F statistics is less than 1. Then the null hypothesis is accepted. Answer is option A. Then if the mean of a Poisson distribution is 5, then the standard deviation of the distribution is. So in Poisson distribution, mean is equal to variance. So 5 is equal to 5. Standard deviation is square root of variance. So answer is root 5. Answer is option B. Then next one. Lactimeria acts as a connecting link between fish and amphibia. Answer is option C. Then in which of the following animals secondary palate is not found? So secondary palate of mammal, it separates the nose from the mouth. So which animals doesn't have secondary palate? It is the amphibians. Answer is option A. Then select the correct match of the larval form with their groups from the following. So here the answer is Brachyolaria, it is Asteroidea. Cysticercus is Cestoda, Planula jellyfish, and Trochophore polychaeta. Answer is option B. Then glycogen synthase, the key regulatory enzyme for glycogenesis, is inactivated by phosphorylation. If you dephosphorylate it, it is in the active form. So answer here is option C, that is phosphorylation. Then next one, which part of the renal tubule does not reabsorb water? It is ascending loop of Henle. Answer is option B. Then protamines, small arginine rich proteins, are characteristically. So they are histone-like proteins found in fish sperm. Answer is option C. Then which of the following moths lack peristome teeth? So it is sphagnum that is lacking peristome teeth. Answer is option B. Then next one, guide RNA dependent insertion of uridylate residue during RNA editing requires all of the enzyme except. So I will brief on RNA editing. I will not give all the points. You have to read it on your own. But some of the basic things, whatever you need, I will be telling for answering this question. So this is the mRNA, that is the target. This is the gRNA. This is the anchor region of gRNA that bind to the 3' end of the pre-mRNA. This is followed by binding of gRNA to the mRNA, gRNA poly-U tail to the mRNA A region, mRNA poly-A tail. So it will bind over there. This will result in a bulging mismatch region of the gDNA that serves as an information template for uracil addition or deletion. So it is, if you take the case of trypanosomes, there is addition or deletion of uracil residues. So this is the first comes the cleavage portion, that is over here. So it will cleave at the site at first unpaired nucleotide of the pre-mRNA target. So cleavage site at first unpaired nucleotide of the pre-mRNA target. So it is cleaved over here, that is the pre-mRNA is cleaved over here. Then depending on the information encoded in the template region of the gRNA, uridylates can either be added to the pre-mRNA by terminal uridyl transferase, that is TUTTase. It will add uridylate to the 3' end of the 5' fragment, that is over here. After this particular cleavage, it will be the tutase enzyme, it will be adding the uridylate to the 3' end of the 5' fragment. Then you have got the exouase that is involved in deletion of uracil. So it is over here, that is this extra U, this U that will be deleted. So exonuclease remains unpaired. Exonuclease, that is exouase. It removes the unpaired uracil from this portion. Then after that you will get a sequence like this. So I am not telling all the enzymes that are involved in this action. Okay, that you have to learn. Then this U insertion and deletion, the 5' and 3' fragments of the mRNA get ligated through the action of editosome RNA ligase. So there is this is the ligation reaction where the pre-mRNA after editing is ligated by editosome RNA ligase, that is KREL1 and KREL2. So this is what is taking place. Then guide RNA dependent insertion of uridylate residue during RNA editing requires all the enzyme. So endonuclease, yes, that is required. Here you have got an endonuclease being natted. Then you have got tutase that is needed. 3' U exonuclease that is not needed because guide RNA independent insertion of uridylate residue they have asked, that is addition of uridylate residue. So you don't need the exonuclease action, but you need endonuclease. Then you need tutase that is involved in addition of uracil to the pre-mRNA, and then you need RNA ligase for ligating the RNA after addition of the uracil residue. So the answer here is option C, that is 3' U exonuclease or exouase. That is not required. Then next one. Heterocysts commonly found in cyanobacteria are associated with. So they are associated with nitrogen fixation. Answer is option B. Then activity of which of the following pairs of plant hormones are similar in delaying senescence? So it is auxin and cytokinin. Answer is option B. Then next one. Xeroderma pigmentosum is an autosomal recessive mutation that affects the mechanism of repair of UV-induced thymine dimer formation in DNA. Which of the following enzyme is most likely to be defective? So Xeroderma pigmentosum, it is an autosomal recessive condition, and where does it affect? It affects the mechanism of UV damage induced thymine dimer repair in DNA. So which of the following enzymes is most likely to be defective? So the defective enzyme is likely to be defective is endonuclease. Answer is option C. Then next one. Sickle cell anemia is a heritable disorder where at least one beta globin subunit of hemoglobin A is replaced, forming hemoglobin S. This demonstrates autosomal recessive pattern of inheritance. Answer is option C. Then in the RNAi regulatory pathway, the dicer enzyme cleaves. So dicer cleaves double-stranded RNA and pre-microRNA and results in the formation of small interfering RNA and microRNA respectively. So these small interfering RNA and micro are double-stranded. So in the dicer enzyme cleaves, answer is double-stranded RNA into short strands. Then next one. Which of the following proteins have not been used in genome editing? So genome editing or genes engineering or gene editing or genome engineering is a type of genetic engineering in which DNA is either inserted, deleted, modified, replaced in the genome of an organism. And the core enzymes that are usually involved is CRISPR-Cas9, transcription activator like effector nucleases, that is TALENs, zinc finger nucleases, ZFNs, and homing endonucleases or meganucleases. So MHCD, option D, that is MHC is not involved. Answer is option D. Then the term protein splicing refers to. So protein splicing is a post-translational process facilitated by an intervening polypeptide called an intane for internal protein. This intane interrupts flanking polypeptide called extines. The intine is responsible for catalyzing its excision from the intine concomitant with extine ligation. So protein splicing, it involves removal of intervening protein sequences termed as intine from translated protein, and then joining of the or ligation of the extine regions. That is called as protein splicing, just like intron exon in DNA. Here intane and extine. Then next one, human hair and rhinoceros horn differ in tensile strength and rigidity though both the structures are made of alpha keratin protein. The extra strength of rhinoceros horn is due to the presence of excess number of disulfide bonds. Answer is option D. Then next one. Processing and modification of rRNA needs a class of small RNAs called. It is called snRNA or small nucleolar RNA. They are class of small RNA molecules that primarily guide chemical modification of other RNAs, that is ribosomal RNA, transfer RNA and small nuclear RNAs. So the answer here is option B, that is snRNA. Then next one. The CRISPR sequence are recognized by. So they are recognized by the guide RNA. Answer is option B. How many DNA duplexes will be obtained from one DNA duplex after 5 cycles of PCR? So it will be 2 raised to n. That is the formula to calculate number of DNA copies formed after a given number of cycles. So where here 2 raised to n, where n is the number of cycles. So 2 raised to 5, because 5 is the number of cycles given in the question, and 2 raised to 5 will be 32. Answer is option D. Then initiation of protein synthesis from most eukaryotic mRNA requires both poly A tail and a 5' cap. Answer is D. Then next one. In hybridization experiments, high stringency washing means. So stringency, it is a term used in hybridization experiments to denote the degree of homology between the probe. If you take southern hybridization, it is the degree of homology between the probe and the nucleic acid that is bound to the membrane. So higher stringency means higher the homology. So high stringency conditions are used to detect identical or very highly conserved genomic sequences complementary to the probe sequence. So high stringency washing is low salt concentration and high temperature. So answer here is option A. Then dicentric and ring-like chromosomes may appear after the cells are exposed to. So after the cells are exposed to gamma radiation, you will get dicentric and ring-like chromosomes. Then next one. In bacterial taxonomy, which of the following serves as the golden standard for identification and determination of evolutionary relationship? It is 16S RNA gene analysis. Then in bacteriophage lambda, the factor acting as a repressor that allows the establishment of lytic cycle is. So it is CRO. Answer is option D. And questions from this lytic and lysogenic cycle and the genes involved there have been asked repeatedly in so many state eligibility tests, not only of West Bengal but of other states also. So please go through it before going for the exam. Then next one. Anthracene 9 carboxylic acid is used in studying the disorder myotonia congenita. It acts as a. So it is caused, this myotonia congenita, it is caused by changes or mutations in the gene called CLCN1, which is responsible for shutting off electrical signals from neurons to the membrane of skeletal muscle cells. So a mutation in this gene, it results in increased signals to the skeletal muscles. So specimens exposed to anthracene 9 carboxylase acid inhibit this ClC1 chloride channels and induce myotonia in vitro conditions. So it is chloride channel blocker. Answer is option D. Then next one. Which of the following plant families is characterized by the presence of pappus? So it is Asteraceae. Answer is option B. Then proteomics techniques is used to analyze the characteristic of molecular protein network involved in living cell is termed as. It is called as functional proteomics. Answer is option A. And functional proteomics, it constitutes it is an emerging research area in the proteomic field whose approaches are addressed towards two major targets, that is elucidation of the biological function of unknown proteins and definition of cellular mechanisms at the molecular level. Then next one. Identify the experiment that's unable to detect the lateral diffusion of membrane proteins. So it is X-ray diffraction. That cannot be used to detect the lateral diffusion of membrane proteins. Then next one. Suppose a new drug has been developed to treat diabetes. Now it's need to examine upon the same group of subjects whether the drug is more effective before or after taking the meal. The statistical test that is needed to perform in this scenario is. So paired t-test is the answer. Paired t-test is a statistical procedure used to determine whether the mean difference between two sets of observation is 0. So here you have to use paired t-test. Then next one. During gel electrophoresis experiment, a protein at a particular pH that matches its isoelectric point will. So at the isoelectric point, the overall charge of the protein is 0. So during gel electrophoresis experiment, the protein at particular pH that matches the isoelectric point will not move either to cathode or anode. It will be immobilized. Answer is option D. Then next one. Beta barrel structures cannot be predicted by hydropathy plot analysis. Choose the most appropriate reason behind this phenomenon. So presence of alternating hydrophilic and hydrophobic amino acid residues in the primary sequence of beta barrel. That is the most appropriate answer. So answer is option C. Then next one. During protein sequencing experiments, the peptide bond contributed by the carbonyl group of methionine residue is cleaved by. So cyanogen bromide cleaves the carbonyl group of methionine residue or C-terminal side of the methionine residue. So cyanogen bromide. Answer is option B. So that's it. Please do like, share and subscribe. Thank you for watching.